In this module, we'll discuss the immune mechanisms underlying type III hypersensitivity. This is our third hypersensitivity module, and type III hypersensitivity is mediated by IgG that binds to soluble antigens to form what are called immune complexes. So how are type III hypersensitivities different from type II hypersensitivities, which are also mediated by IgG? And here's an example of that. In type II hypersensitivity, IgG binds to cell-associated antigens, and it induces activation of complement and opsonization and antibody-dependent cell-mediated cytotoxicity. Type III hypersensitivity reactions occur when antibodies bind to soluble antigens. And when those soluble antigens are present at high levels for sufficient periods of time, they can cause the antibodies to form immune complexes with those soluble antigens. Immune complexes happen when antibodies and antigens kind of aggregate together, and this can cause these aggregates to precipitate out of solution. These complexes precipitate out of solution and accumulate in places like the synovial fluid, blood vessel cell walls, and glomeruli. And these, especially in the synovial fluid and the glomerulus, these are filtration points. And so the plasma filtration functions of these areas really promote immune complex accumulation. Complement, when it binds or associates with endothelial cell walls, can induce vasculitis. And this is because it can be very efficient at activating the complement pathway. And so here we have a bunch of immune complexes that have bound to this capillary cell wall. They've recruited C1, and this leads to potent activation of complement. So for example, you get the formation of C5a and C3a. C5a and C3a in turn can induce vasodilation and recruitment and activation of neutrophils, which then can release cytotoxic granules, reactive oxygen species, and mediate necrotic death of some of these endothelial cells. And this is what leads to the classic symptoms of type 3 mediated disease, which is vascular necrosis and vasculitis and rash and fever. It should be noted, the ability of these complexes to activate complement is profound. And so often complement activation of these by these complexes can lead to C3 consumption, such that you don't have very much C3 left in the serum because you've used it all up. And this is actually a diagnostic marker for some type 3 diseases. Complement, besides inducing vasculitis, can also induce joint inflammation. So these immune complexes have activated complement within the joint, leading to arthritis and arthralgia, and they can lead to kidney inflammation and glomerulonephritis, leading to proteinuria and kidney dysfunction. Complement, or susceptibility, to type 3 hypersensitivity is linked to genes that regulate a variety of functions, but those include immune tolerance, as well as genes and particularly components of the complement pathway that are important for normal clearance of immune complexes, as well as scavenger genes that are important for removal of necrotic or apoptotic debris. So the reasons for why some people are more susceptible to immune complex mediated disease are not totally clear, but we do know some of the genetic linkages that may help explain those susceptibilities. So what are the hallmark signs of type 3 hypersensitivity? Well, we've just discussed them. They're vasculitis or rash, arthritis and arthralgia, glomerulonephritis, and signs of systemic inflammation, including fever, are common. So one key type 3 hypersensitivity reaction that you're going to want to know about is serum sickness. And serum sickness is a response to foreign antibodies. So let's say you go hiking, get bit by a snake, go to the hospital, and they're going to treat you with an antivenom. This antivenom can come from injecting snake venom into a horse or sometimes a goat, and leads this horse to produce large amounts of antivenom antibodies, which can then be injected into the patient to ameliorate the effects of this snake bite. The problem with this is it can also lead to an immune response. These are foreign proteins, And there are B cells that we have that could potentially bind to and be activated by these foreign proteins. And so here we now have these anti-horse immunoglobulin B cells that with T cell help can induce an anti-horse Ig response. And these IgG molecules that are induced by this response can then form immune complexes with these horse immunoglobulins. And this leads to the classic symptoms of rash and joint pain and fever. Typically, these types of reactions will occur 10 to 21 days after exposure, because that's about how long it takes to get this really potent anti-horse IgG antibody response. There's a number of ways to treat this besides not injecting any more horse antibodies into the patient. NSAIDs and antihistamines and corticosteroids could all potentially be helpful. And this is a very painful experience, per my understanding. I've shown a couple of pictures of the type of rashes that are induced during serum sickness. And there are other components or other substances that can also induce serum sickness, certain non-human monoclonal antibodies, as well as antithymocyte globulin. You can also induce a serum sickness-like syndrome with drugs, and this can be induced by antibiotics like penicillin and cephalosporins, which is relevant to this class. And again, these serum sickness-like syndromes can occur 10 to 21 days after drug treatment, and they occur when these antibiotics or drugs are able to induce an IgG response, and that IgG is able to form immune complexes with the drug. And so drugs can induce a variety of hypersensitivity reactions, including type 1 hypersensitivities, which we've discussed previously. So this is penicillin, and here we have a mast cell that's coated with penicillin-specific IgE molecules. Drugs can induce type 2 hypersensitivities, and so here we have a penicillin that's covalently attached to the surface of a red blood cell, and penicillin-specific IgG is leading to red blood cell destruction. And drugs can lead to type 3 hypersensitivity reactions. So here we have penicillin that has formed immune complexes with anti-penicillin IgG antibodies, and this can induce penicillin serum sickness-like syndrome. Another classic type 3 hypersensitivity disease is systemic lupus erythematosus, or SLE. And this happens when we develop antibodies to a variety of nuclear proteins, including ribonuclear proteins. These antibodies are sometimes called Smith antibodies. We develop antibodies to double-stranded DNA, as well as kind of non-specific antibodies, what we call anti-nuclear antibodies, to a variety of nuclear components. These are kind of a non-specific diagnostic marker. Anti-nuclear antibodies are present in lupus, but also in a variety of other autoimmune diseases. And the result of immune complexes that form from these antibody responses can have a variety of effects. For example, you can have involvement of the skin, where you get a malar rash or a discoid rash, or develop photosensitivity. And so here I've shown an individual with kind of a classic malar or butterfly rash on the cheeks. Not all people develop this, but it is considered kind of one of the hallmark signs of lupus. And you can develop these kind of discoid rashes as well. You can have effects in the kidney, including glomerulonephritis and proteinuria. You can have other effects such as migrating arthralgias, which are common, effects in the mucosa and the serosa, neurological effects. And the onset is mostly in women of their childbearing years. And the most common way to treat this is probably corticosteroids, but the treatment that someone would undergo would depend on disease severity. And the symptoms for systemic lupus erythematosus are very variable. Even the presence of autoantibodies is variable. In fact, these autoantibodies can be present on one test and not present a few weeks later. And so it can be a very tricky, difficult disease to diagnose. So here is a summary of the mechanisms underlying type 3 hypersensitivities. Take a moment to review to make sure that you're gleaning all the information that you need to from this lecture.